Hi students, welcome back to DNA repair. In the previous session, we had mentioned about the double-stranded break repair, which normally happens because of the exposure to ionizing radiations. And we found that these ionizing radiations can result in the formation of double-stranded breaks in various double helix. And usually there are mainly two options by which we can overcome it. One is by a non-homologous joining method, and the second one is a homology-directed recombination. Today, we are going to talk about the non-homologous joining method, and we can see that in this figure, if a double-stranded DNA gets exposed to radiations, sometimes it might result in the formation of various blunt cuts. At the same time, you can also see unequal cuts of the DNA could also happen. And if you're having a blunt cut, the chances you can simply just join them together, irrespective of checking whether they are homologous or not. That would result into the condition known as a non-homologous joining method. At the same time, if you're having an unequal cut also, sometimes the overhangs will be processed by a process called the N processing, and by which sometimes nuclease actions will remove it, or they might be trimmed, or sometimes DNA polymerases can add few bases also onto it, depending upon which is compatible. And ultimately, these two ends are being joined together, and that is also referred to as non-homologous joining. But in either of the cases, if you look into the figure which is here, you can see that large amount of DNA is being lost in both the type of non-homologous joining method. And gene or DNA, as we know, is very much important. And any loss of genes or DNA fragments within it would result in formation of errors during the survival of the organism which is being exposed to such radiations, or the cells get critically affected. So, non-homologous end joining method results in a error-prone type of replication even after the repair mechanism has been carried out. Many species of bacteria, including Escherichia coli, they lack an enjoining pathway, and they mainly completely rely on homologous recombination to repair the double-stranded breaks. But we can see that non-homologous enjoining proteins have been identified in a number of bacteria such as Bacillus subtilis, Mycobacterium tuberculosis as well as Mycobacterium sphygmatis. And in Mycobacteria, the non-homologous enjoining is much more error-prone than yeast. And the bases are often added to as well as deleted from the ends of the double-stranded breaks during this repair, as I had mentioned earlier about the end processing which could be done. Usually in the case of bacteria, this mainly happens in the conditions where the bacteria has been exposed to high levels of desiccations. As we know, even ionizing radiations brings about some desiccation towards the bacteria. And to overcome this, mainly two proteins are mainly involved in the bacteria for the non-homologous joining. One is a Ku homodimer, and you also have a ligase D or the Lig-D, which is it is abbreviated as Lig-D, which is a multifunctional enzyme which is having ligase, polymerase as well as nuclease action. So let's just have a look into that. Into the case of prokaryotes, you can see that they could have a double-stranded break when they have, at the end of it, you have the Ku proteins which are being bound to it, and further the Lig-D or the ligase D, which is a complex having three main functions such as the nucleases, ligase as well as the polymerase function. That the single enzyme with it's a multifunctional enzyme, okay, a single enzyme with different functions, that itself will do as a nuclease. It does a nuclease action if it needs to process the ends, It does a polymerase action if it has to synthesize a new DNA strand, new DNA fragments over there, or it does a ligase function if it has to seal the nick, ultimately leading into the sealing of the double-stranded breaks. Now, coming to the eukaryotes, they utilize a number of proteins which participate in the following binding of Ku heterodimer to the double-stranded DNA ends, and they also have the recruitment of DNA protein kinases. Uh, this is referred to as MRX complex in the case of yeast. And the next, it will have the processing of ends, the recruitment of DNA ligase 4, XRCC4 complex, and which brings about the ligation. And you can see that in the coming figure, it's showing. You can see that. You have a heterodimer of Ku protein which is binding here. And in the case of yeasts, the complex which does the process of end processing is referred to as a MRX complex. Whereas in mammals, it's a different set of proteins which act here. Then after that, a gap filling happens by the polymerase enzymes. The different polymerases are being involved in yeast as well as in mammals. Further on, ligation happens. That's what we can see. And ultimately, it will result in the formation of a break-free double-stranded DNA in either case of Saccharomyces as well as in mammals. But the thing that we should note over here is, regardless of what are the processes, what are the proteins which are being happening in either of all these cases, they have an end processing, and then they have a gap filling by the polymerase action. Then finally they have a ligation so that the double-stranded break is being repaired. And we need to keep in our mind that in non-homologous end joining method, whether let it be in prokaryotes as well as in eukaryotes, some amount of DNA is being lost, and that would result in the error prone repair of DNA. And usually non-homologous end joining doubles repair of the double-stranded breaks, it happens at all stages of the cell cycle, bringing about the ligation of two DNA double strands without the need of a sequence homology, and as a result, it has been found to be error-prone. I hope you got a better idea of what is non-homologous end joining method. I'm not going through the details of each and every protein because my aim was to give you an overview of the non-homologous end joining method, which happens in the prokaryotes as well as in the eukaryotes. And it is a method which helps them overcome a double-stranded break. I hope you are clear with this. With this, we come to an end of the non-homologous end joining method to repair the double-stranded break repairs. Thank you.